Hi, welcome back. This short lecture will be talking about acquired immunity or specific immunity, and we will be talking about B cell activation and antibodies and immune responses, which sort of sets us up for the basis of vaccinations. So let's go to the first slide, which is introduction to the idea of acquired or specific immunity. This is learned. The body is exposed, the immune system is exposed to a particular antigen, it responds to that antigen, and it remembers it. So keep in mind that all blood cells, both white blood cells and red blood cells, begin their development in the bone marrow, rising from hematopoietic stem cells, and B cells, B lymphocytes, go on to complete their development in the bone marrow. The T cells leave the bone marrow immature, circulate, and then go to the thymus. So T for thymus, which is where the T cells complete their development. So next slide is looking at both B cells and T cells. Both of these are involved with specific immunity. Both B cells and T cells have a specific antigen receptor, and it is incredibly specific. And interestingly, those are generated in the bone marrow during development randomly. The genes that code for the protein antigen receptor on the surface of the B cells and T cells, those genes go through scrambling and mixing of different segments randomly, and the end result is that they generate new versions of the gene that code for different proteins. So in the figure below, these antigen receptors shown on the surface of the cell, in this picture I've shown 3 different antigen receptors. This one on the left has little circles on it, so it would presumably bind little circular antigens that would bind to that. The middle cell has square receptors, so it would presumably bind some sort of square-shaped antigen. And likewise, the cell on the right has sort of V-shaped receptors, so it would be binding sort of V-shaped antigens. That's the idea. And these receptors are generated randomly. No plan as to what might be needed. But since you literally have tens of trillions of different cell possibilities, the chances are real good that you'll have something that will bind whatever comes along. Tens of trillions is an unimaginably large number of possibilities, and that just came from the rearrangement of the genes during the B cell and T cell development. Both B cells and T cells become cloned during an immune response. So let's go to the next slide and take a look at some of this. So again, we're talking about B cells and T cells, and both of those are cloned during an immune response. And cloning makes active cells, sometimes referred to as effector cells, and memory cells. And it's the memory cells that stick around and give you immunity later in life to the same disease. So what the picture is showing is both a primary and secondary immune response. So I'm going to go through this after this slide with one that I'll draw. But here's the basic idea. It's a little bit small on the figure, so I'll enlarge it when I draw it later. But this is the primary immune response right now. So primary just means the first time the cell encounters that particular antigen. It finds one T cell or B cell, it's said to be naive, meaning that this cell has never seen that antigen before. It just happens to have a receptor on the surface that recognizes the antigen. When those two bind, that activates the lymphocyte to begin dividing, and it makes clones of itself. So it divides once at the beginning, and then these two lineages of cells produce two different populations, and we have short-term effector cells and we have memory cells. So the two populations. So that's after the first response. So now we have many B cells and T cells. Some of those will fight the infection today, and others will go off and lie quiet, but ready to fight the infection, the same antigen, if it sees it again. But now we have lots of different clones, all now with the same antigen receptor. So we started with one single cell that just happened to be able to recognize that antigen, and now we have many cells, both long-term memory cells and short effector cells that will fight at the moment. The clones all have the same antigen receptor. So for the secondary response, meaning the second time the body comes in contact with the same antigen, now we have a whole army of cells that are available with the same antigen receptor to bind the same antigen. And now we can have a much larger and much more rapid immune response after the body has become sensitized. So the next figure I want to draw is looking at B cells in particular. And B cells, remember, when activated, turn into plasma cells that produce antibodies. So that's the story that I want to talk about. So let's go to a blank page here. And here's the idea. In the lymphatic tissue, we have billions of different B cells. So I'll just try to keep this somewhat clean, and let's just say that here are maybe 3 different B cells. Maybe they're in the spleen, maybe they're in the lymph nodes. Doesn't matter. They're lymphocytes, they're in lymphatic tissue, and they have, each one of these has a different type of receptor. So let's say this one has little half circles on it. And again, this one maybe has little squares on it. And I'm limited only by my imagination and geometric shapes that I can draw. So this has V's on it. You get the idea. Each of these little shapes on these antigen receptors is representing a uniquely shaped protein. So those are the AG receptors. The point to appreciate is that they're all different. Circles, V's, or squares. And you literally have billions of those, all with different antigen receptors. And they are naive. They've never seen an antigen that will bind to their receptor until, let's say, here comes COVID-19. COVID-19 is rather large because I have to be able to write in it. So there's our COVID-19 virus causing the pandemic that we're living through now. And It's a coronavirus. You may have seen electron micrograph pictures or artists' pictures, but it has, the way it got its name was it just has all these little spiked proteins on the outer surface. Looks like a little crown or a little halo, so it's referred to as a coronavirus. The original SARS virus back in 2003-ish was another coronavirus. This one's very similar, yet different in some important ways. So there's the virus. It gets into the body, and the immune system cells are bumping into it, and by chance, eventually, hopefully, the virus, in addition to causing trouble in the respiratory system, also winds up binding to one of these B cells. So let's redraw that. So if I can give myself a little bit more room, here is B cell again. And it just happens to have a little shape on the antigen receptor that binds this particular antigen. So COVID-19 binds to the B cell. Here's the virus in red. And there are other steps. T helper cells are involved. It's not as simple as I'm drawing it, but this activates the B cell. So binding the antigen along with other signal chemicals and the help from T helper cells leads to activation of the B cell. And then the B cell divides. And it divides into two daughter cells that give rise to two different populations. So one population of clones from this particular individual B cell make lots of B cells, and these are going to be the memory cells. And what's important is that all of them are genetically identical to the original cell, and they all have the same antigen receptor on them. So all of these have these little half circles, and I'm only drawing a couple in the interest of time. But these are the memory cells. They're identical. Memory cells, let's say all with, I'll abbreviate with, same antigen receptor. So that's one population. Those are the memory cells. They won't fight the infection today. Hopefully they will stay with you, and the next time you come in contact with COVID-19, they will be ready to go. And now you have maybe millions of them instead of just one. The other population of cells that are produced develop into plasma cells. So you have memory cells and plasma cells. And the plasma cells, if you remember, release antibodies. And antibodies we will abbreviate as AB. And what's important is that the antibodies look like the original antigen receptor. Now, I drew them on the B cell in a simplified form as just a stick with a U at the top. But the antibodies actually are a little bit more complex than that cartoon drawing that I made in a hurry. But I didn't want to do this on every B cell. That would have just bogged down your note-taking. But what's important is that these antibodies have the same antigen receptor. So this is the antigen binding site of the antibody. The little half circles. Antigen binding sites. There are two for antibodies. They have two arms, one on each side. So what happens is that now you are producing tremendous numbers of these antibodies, lots and lots of them. And they go into the blood, into the plasma. And they circulate in the plasma, in the blood, and whenever they find COVID-19, COVID-19 is making you sick, and COVID-19 has these little antigen proteins, part of the virus. And now the antibodies come along. You can see what's happening. The antibodies come along and they bind to it. And maybe here's another antibody that comes along and binds to it. The arms are somewhat flexible on the antibodies. And pretty soon they cover up the antibody, or excuse me, they cover up the virus and renders it useless and renders it, it sort of tags it for destruction, opsonization again. So tags for destruction. And so these are the circulating antibodies. They can only bind things that are outside of cells, circulating around in the plasma, or wherever they're secreted. So that's the story of B cell activation. I hope that it makes sense. So just to review, lots of B cells, all naive, never having seen a particular pathogen. Along comes a particular pathogen that happens to be able to bind one of those B cell antigen receptors, causes that B cell to become activated, makes clones, memory cells, and plasma cells. Plasma cells produce the antibody, and the antibody has the same exact antigen binding site as the original B cell. Those antibodies then circulate in the blood and bind to the original, the same antigen. The next slide looks at the immune response. So the print is a little bit small. All three of these figures are showing basically the same idea. As I mentioned in the text, I just went to Google Image, typed in immune response, and this is what I get. So what we're looking at is both the primary and the secondary immune response. As determined by antibodies in the blood. by antibody titer. And titer just means concentration. So if we're talking about the titer of anything, we're talking about the concentration. So we're just talking about the number of antibodies in the blood, in the serum. So the bottom left figure, this might be the easiest one to start with. So note the scale. We're talking about weeks across the bottom, so 0, 2, 4, 6, 8, etc. And at this point in time is our first exposure to the antigen, to the pathogen. So first exposure. And notice that, let's just follow the red line. There's a bump, and this is the primary response. This figure is talking about antigen A and antigen B. We can forget about antigen B and just look at antigen A. Just the red line is all I want to focus on. There's a period of time before the antibody titer starts to increase. And it's often a little bit longer than what's shown. But there's a lag period in time. So this is when, during the first exposure, this is day 1 when you became exposed to the pathogen, or the first day that you received a vaccination. It's the first day that the antigen went into your body, and it wandered around, and it took some time for it to find the one B cell that could bind to it, to become activated, and to start to produce antibodies. So there's this little latent period, this lag period. And then you start to see the antibody titer going up. And it's no units here on the side, but the level is going up. And after a couple of weeks, we hit the top of the red line. That's the maximum number of antibodies that were produced from this immune response. We bound to one B cell, made clones, made plasma cells, made antibodies, great, and then that sort of tapered off. And then the level of antibodies in the blood now, though, is higher than it was. Before, it was 0. Now it's a little bit higher. So we have a little bit of protection inside our blood from the antibodies. Now, if we are exposed to the same antigen again later in time, about 7 weeks or so on this drawing, we have a secondary immune response. And now the reaction, the antibody increase is much more rapid. We don't have that lag period in time because we have memory cells that are already present. There are now millions of cells of the same antigen receptor to recognize the same antigen, and that activates many plasma cells, and we have this very large increase in the antibody titer. And then after a period of time, that drops off again. But now, note that our antibody titer is higher than it was before. So now we have a higher concentration of antibodies. That's the idea. The figure on the top is a little more dramatic. A couple of things to note. This is the primary response in terms of antibody titer. And this is the secondary response. And they're not showing the little arrows on when the exposure to antigen occurred, but that's okay. Notice that after the primary response, antibody titers do not go to 0, which is what I don't really like about this figure here. It looks like they're going almost to 0. But not to 0. and then you are exposed for a second time, and now our antibody concentration is much higher. And that can last for quite a while. And this is what vaccinations do, is that they give you protection from the pathogen, from the virus, by activating your immune system ahead of time so that you have this large concentration of antibodies in the blood to bind the virus when it enters the body before it can infect you and before it can cause disease. This is what's in the news right now when they're talking about doing serologic testing. So the buzz at the moment is trying to identify people that are now resistant to COVID-19. And the way that they're doing that is to take a blood sample and measure the antibody titer that's in their blood. And if it is high enough, you have a high antibody titer, then it's presumed that that will give you protection. Serologic testing for, let's say, antibody protection. And if you know that you are protected and that you're now immune from the disease, the thought is, well, you can go back out into society and you're no longer at risk for infecting anybody else. There are some questions, though, that still have not been answered that need to be. One is, how high does this titer need to be? Don't know yet. And how long will it last? Don't know that either. And you can make all kinds of assumptions. Some of those may be more valid than others, but the point is, right now we don't have data to support that, but we're working on it as quickly as we can. The other topic, of course, that's in the news is a vaccine for COVID-19. They want to take COVID-19 and break it up into little pieces and develop a vaccine for it. And then you can inject fragments of that virus that will bind to immune system cells and make antibodies to that fragment. Everybody wants it. Well, I don't know about everybody. I don't know where the anti-vaxxers stand on this issue at the moment. But the next, or later on in this series of immune lectures, I'm going to talk a little bit more about vaccines. I hope that makes sense. And we will talk about antibodies next. Take care. Bye-bye. Stay safe.